So first of all, welcome. It is a pleasure to be here. Most of the teams with collaborators, shareholders, and other fellow members of the Royal Society of Medicine. We are here to talk about Algocyte Proxima, which is the device that we are launching officially today, and Algocyte in general, our vision to the future.
Thank you, Hector. It's my pleasure to be here and share this session. We have 3 talks today that they are closely connected. We begin with immune system, and then we move to the causal system medicine and causal AI, and then finally we move to the disease signature for precision healthcare. With that, it's my pleasure to introduce our first speaker, Professor Kourosh Saeb-Parsy. He is a professor of transplantation at University of Cambridge, consultant transplant surgeon at Addenbrooke's, and chief medical officer of Algocyte. His work connects transplantation, cancer biology, regenerative medicine, and immunotherapy. He will open the session with his talk, A System That Rules Them All, the immune system in health and disease. Please join me to welcome Professor Kourosh Saeb-Parsy. Glory.
Thank you very much for the invitation to speak to you today. It's a genuine pleasure. I've had the pleasure of working with Hector and the rest of the team for many years. So I am both personally and professionally delighted to be here today at such a fantastic venue amongst colleagues and friends. The immune system has been something that has taken a significant part of my professional career over the last couple of decades. I'm a transplant surgeon, so almost every one of my patients have disruptions of their immune system. And of course, this is in the context of the rejection of an organ transplant. But I've spent the last many years really trying to understand how the immune system regulates abnormalities, be it in the context of transplantation or cancer or cellular therapies or infections, and how we can understand and monitor this. I'll start with a bit of a personal story. When I was a very young, I was just turned 18, medical student, arrived in Cambridge to read medicine. And really, that was the first time I started thinking about the immune system and how we monitor the immune system. And at that time, it seemed very strange to me that the way we monitor this very complex system is to basically categorize our assessment of it into a binary system of right or wrong. And it's taken me about 20 years or so to really get to a point where I feel that we are on the cusp of actually making a contribution to actually breaking this mold. I'll tell you more about this story and how my now 16-year-old reacted to the same facts. So, the immune system. It's undoubtedly probably one of the most complex physiological systems that we have. It has a very comprehensive series of components which take the shape of different cells, but also a variety of releasable factors. And this very complex network has evolved over millions of years to protect us against primarily invading organisms. But as you'll see, it is now very well established that actually this system is not only impactful in the context of infections, which is how the system evolved, but also actually protecting us against deviations from normality within our homeostasis, within our bodies. What I'll do is I'll go through 3 or 4 slides, just giving you examples of how the immune system is impactful in contexts that historically we were perhaps less aware of. Of course, we know the immune system is important in infections and in vaccines, bacterial infections, viruses, fungal infections, and so on. Also, autoimmune diseases such as diabetes, rheumatoid arthritis, that's not a surprise. But actually, we are now increasingly recognizing that the immune system has really important roles in things like cancer, neurodegeneration, mental disorders, but importantly also normal function and recovery of our physiological systems from pressure, from exercise and beyond. So my patients are pretty much all immunosuppressed, with the exception of those that have very rare transplants from genetically identical individuals, so from twins. So if you had to guess, what is the likelihood of somebody with a kidney cancer actually getting with a kidney transplant getting cancer after about 20 years of transplantation? The likelihood is almost 100%. It's almost guaranteed that they will get a skin cancer. The reason for that is that we know that the immune system is constantly identifying abnormal cells and eliminating those abnormal cells. And actually, if you suppress that ability of the immune system, then tumors and cancers are much more likely to arise. And of course, that's the basis of the AIDS-defining cancers as Kourosh sarcoma. But it's now increasingly recognized that actually not only we can leverage understanding the immune system to prevent cancers, but also cancer therapeutics. And you would have heard of things like checkpoint inhibitors, where we can augment the ability of the immune system to eliminate cancer cells. And modulating and impacting the immune system in order to develop therapies for cancer is now a very exciting emerging area. What about cardiovascular disease? It's now very well recognized that our likelihood of us being afflicted by cardiovascular diseases such as myocardial infarction, so heart attacks or strokes, are very high. And I'll show you some data on this. And it's now clear that actually when you have disruptions in our system, the immune system is one of the key factors that determines what the consequence of those disruptions are and why people with similar risk factors sometimes actually have events such as myocardial infarction and others don't. What about neurodegeneration? So if we think of something like Alzheimer's disease, as you know, this is probably one of the epidemics that is affecting us and will continue to do so over the next few decades. We have no real prognostic markers. We can't really determine who is likely to get Alzheimer's or similar conditions, and we have no treatments for it. But it's now well established that the functioning of the central nervous system, including in cognition, is very much affected by disruptions and disturbances in the immune system. What about normal day-to-day function? Sleep. So it's probably been known for, millennia that sleep is important, right? If you look at mammalian species, the one thing that they all have in common is that actually they sleep. And we've always assumed that actually the role of sleep is recuperation, an ability for our both central nervous system, but also our peripheral physiology to recover from stress, from deviations from normal. It is now clear that the impact of sleep on other functions is mediated at least in part through the immune system. Actually, it's direct linkages to things like depression, so mental disorders, but also healthy functioning, resistance to infections. All of these are most likely to be mediated through disruptions of the immune system. The effect of the immune system is evident at every stage of our physiology. So during aging and, of course, pregnancy, but also is a key determinant of our normal physiological status when it comes to things like mental ability or physical performance. So the fact that the immune system is a key component of determining how we function and how that deviation of function results in pathological states is now clear. The question now comes is how do we leverage our understanding to be able to apply a therapeutic lens to what the immune system does. So I'll show you a complex slide which sort of maps what we anticipate will happen or how we will start thinking about the immune system in the future. So we start at the left-hand side corner here. So the idea here is that there are a number of parameters that will affect the nuances of the functioning of our immune system and our immune cells. And that the idea is that these will have an impact or determining the immune status in a particular individual. And I'll come to speak about this more. So that actually it determines the set point of each of our immune systems. And these basically determine what we call immune phenotypes. So the idea here is that actually my immune system or my immune phenotype is affected by a variety of genetic and environmental factors, and that these are actually specific to me. And deviation from that status for me has consequences for us in terms of disease status. Now, the holy grail here is that by being able to map our immune phenotype at very high levels of detail, and by leveraging the power of machine learning and artificial intelligence, we will be able to identify these immune phenotypes at ultimately, if not in individual level, in small groups of patients, and that then these will be predictive of our responses, both during normal physiological function, so during aging, through exercise, both physical and mental, but also response to treatment. The problem with this model, or not the problem, the challenge that this model poses is that this is all very futuristic. What are the likelihood of us being able to actually map that onto Mrs. Jones, who is waking up in the morning and goes to work? Right? This world seems many years, if not decades away from actually really being able to therapeutically leverage our understanding of the immune system. What I'd like to show you is that actually we're not that far from being able to do this, but there has to be a very specific strategy for us to be able to convert this complex system into something that's actionable in the very near future. So let's look at some of the key cell types in the human system. So these are different types of immune cells. These are what we call violin plots. So what you see on the left-hand side is basically a scale of abundance, how proportionally, whether they are a high percentage of the immune cells or low percentage. What you can see is that if you would take any one of these cells, some patients have a lot of these cells, and some patients have very little of these cells, right? So why is it that when you look at, for example, CD4 T cells, these are a subset of lymphocytes in the blood, why is it that some people have this particular cell at very high abundance, and in some other people, these cells are at a very low abundance? And what I should say is that actually this is the variance or the repertoire of these cells in completely healthy individuals as far as we can determine. So these are not patients that have got particular diseases or disorders. This is just what their biology, their individual biology is. So the idea is that if we were to understand these, how these different cells map onto the population, what we might be able to do is to start with a sort of what looks like a very heterogeneous population and be able to say, actually, there are a small subset of people, healthy individuals, that actually share a particular phenotype or a particular sort of aggregation of these cells. And this is what we call immune phenotypes. The critical thing here is that, first of all, the normal population has a very diverse range, basically in every category that we look at. So whether this is the number of a particular cell, the abundance of a particular protein, or a chemical, a messenger molecule that we call cytokines, there's huge variations in the population. This variation exists even if we try and select people who are very much alike. So if we were to take a particular population and we try and control for things like race and age, we still find that there's this very diverse range. So my first point is that this binary categorization of saying that actually if you have this particular percentage or number of these cells, it's normal, and if it's higher or lower than it's abnormal, this binary categorization is oversimplistic. And that's in fact what medicine has been doing with analytes, anything that we measure, for basically over a century. Normally we measure something and we put it into a normal or an abnormal category for the individual person. So that is very oversimplistic. Let's take a little bit, look a little bit more at one particular subset of cells. What you've got here is a study that was done again, selecting for people who were otherwise completely healthy, no known medical condition that would affect the immune system. What you have is you've got age along the bottom, and each color here is a different individual. And you can see that the colors are sometimes linked, and sometimes the colors are going up, sometimes they're going down. So what this, for each individual, what we're seeing is what happens to the number of that particular cell in that individual over time. All right, so we are now taking individual people, completely healthy, we're measuring this particular cell, and then we're looking at what happens over a period of time. As you can see, is that actually for some people, the number of these cells goes down over time, some people it goes up, some people it's static. So the authors of this particular study used this and a number of related matters to basically try and predict the likelihood of dying over the next period of a few months. Now, you would think that for completely normal people, the strongest predictor of dying is age, right? That seems like a reasonable assumption to make, particularly when you're looking at people who are sort of in their 60s, 70s, and 80s. But you know what was even a stronger predictor of dying? It wasn't actually their age, not their chronological age, but what they described as their immune age. So in fact, they were able to come up with a collection of parameters that they said defines the age of their immune system, so their immune age, and that actually this immune age was a good predictor, a better predictor of the likelihood of them dying over the next few months than it was that their age alone. Now, so this is what happens to these patients. These patients are more likely to die over a period of about 2,500 days. Actually, this is not a particularly long period of time, right? So it's just about 5 to 6 years. Now, you might quite rightly say, well, maybe this is a quirk of this particular study. Is this reproducible? If other people start looking at our immune system, do we find that actually the immune system is a predictor of wellness or being unwell in patients that are otherwise have no known clinical conditions? So here's another study. Again, without going into the details, they did a similar study looking at the total number of T cells. T cells are one of the main lymphocytes, and then different type of cells, neutrophils, that you'll hear us talk about, CD4 T cells, and so on. And basically, what they looked at is what is the likelihood of dying, of a patient dying based on the number of these different cells that they've got. So, for example, if you have a particular range of neutrophils, then your likelihood of dying is about 36% higher compared to some other people. So just the absolute number of these cells in your blood, or in the blood of these patients, have a very significant, statistically significant chance of predicting whether that person dies or not. Remember, these are not patients; these are otherwise healthy individuals. So this idea that immune cells, are at least predictors of death, it's now robustly confirmed. Now, I call them as predictors, right? Because these studies are currently correlation studies, right? So we are, because these are observational studies, we're not really showing a cause and effect. So it could be that there's some common factor that affects both risk of dying and the immune cells. But the idea that there is a strong correlation is fairly robust now. Some of these cells here are not the type of test that we measure or predict in a routine manner. What do we mean by that? So we actually measure neutrophils very frequently. I'll come to that in a second. But things like NK cells are natural killer cells. They're a particular type of lymphocytes. Actually, very few people have their natural killer cells measured. So a challenge that arises with the story that I've told you is, well, for us to really understand the immune system, you have to do very deep, very detailed analysis of the immune system. And actually, this is maybe not actionable on a population scale. So I want to zoom out now and say, look, what is the simplest and most common methodology of actually looking at the immune system? And that brings us to what we call the full blood count, or the complete blood count, as is known in the US. So the most common medical test performed is this complete blood count. If anybody in this room has had a blood test, it is very likely that this test would have been part of the test that you had done. It's done about 4 billion times a year. So it's the most common medical test, full stop, not just blood test. So the vast majority of people at some point will have this blood test done. Now, the current way that the healthcare systems have these tests performed is during a period of being unwell. So doing the test is a response to having some symptoms or some abnormality. So what you've got here is the different components of these, and I'll just pick 3 of them. So WBC is white blood cell counts. That's the total number of your white cells, and there's different component white cells as I showed you. And then some of these components are neutrophils that we talked about, and lymphocytes. And these are normal reference ranges for men and women. Let's pick the total white cell count. So for males, this is the same for males and females. So the reference value is between 4 and 11. So what this means is that when I go to, I've just come from Cambridge, if I go to my clinic, which I'll do on Thursday, what will happen is if one of my patients has this blood test done, if their value of white blood cell count is 4, they're normal. If that person's white blood cell count 3 weeks later is 11, that's still normal. So I want to take you back to the story with my daughter. She's 16. She was 14 at the time. I said, what do you think your doctor will do if your white cell count, she just started learning about white cells. What do you think your doctor will do if your white cells have gone from 4 to almost 12, so almost 3-fold? What will your doctor do? She said, there's something wrong with me. The doctor will want to know what's wrong with me, what investigations I'll do. I said, no, actually, your doctor will ignore it because you're still within the normal range. You see that this mindset of binary thinking is completely incongruent with our understanding of not just the immune system, but actually physiology. It seems, I think, logically implausible that if a key component of our immune system is deviating by 300%, and that does not have is either not a significant response to a perturbation or has consequence. There is no other physiological parameter that can go up 300% and we would ignore it. So blood pressure or temperature or heart rate or respiratory rate. There's nothing else that would tolerate such a wide variation. So this is a test that we do all the time, with the caveat that we really only do it when somebody is abnormal. So the trigger for doing this test almost always is a symptom or a medical procedure or an operation or something that has triggered that test to be done. So of those 4 billion tests that I mentioned, very few of them, and I'm going to be deliberately controversial, I'm going to say none of them, 0% to 4 decimal places, are done on healthy patients because we just don't do them. So imagine now that I go see the doctor and I'm feeling unwell, either because I've got some symptoms or some signs, and then my doctor measures, does a white blood cell, does a full blood count, and I have a particular value of neutrophils or white cell count. It is almost impossible for the doctor to know whether that value is normal for me or not. All they can do is to say, well, is this within the normal range? It may be that for me, the value is actually 4, and when I see my doctor, the value is 11. It's massively abnormal for me, but the doctor won't be able to tell me that. So, does it matter? So what I want to do is just show you a couple of examples of studies that have been done to show that actually this indeed matters. So this is a pivotal study done across 2 centers where, again, they looked at completely healthy individuals that were being followed up by their family physicians, by their GPs in the UK and New Zealand. These patients, people were completely healthy, had no ongoing medical symptoms, but also they didn't have any predisposing factors. So smoking, diabetes, hypertension, completely normal. And what they did was they just measured their total white cell count. That's the total number of all of their white cells added together. And then they monitored them over a period of time. And what they found is they calculated this thing called the mortality hazard ratio. What this means is how likely is that person to die after a period of time. Notice that actually this goes from 1 is basically it has no impact. That's your sort of control. So if your mortality hazard ratio is 16, that means you're 16 times more likely to die. If it's 0.5, that means you're half as likely to die. And as you can see, is depending on what the total white cell range was, then the mortality hazard ratio was very variable. So it was as high as 16 times more likely if the white cell count was really low. But if it was very high, it was also 8 times more likely. But I want to draw your attention to this pink box. This pink box are the people whose total white cell count was within the normal range. So these are the people that the doctor would say, don't worry about it, your white cell count is normal. Even within that range, we have a hazard ratio that goes from under 1, almost 0.25, to almost 3 times. So this is almost a 6 times increase in likelihood of dying, just simply based on your total white cell count. One other study. So in this particular study, they also looked at the ratio of the 2 of these cell types. So the neutrophils, so these are part of what we call the innate immune system. These are cells that are very first responses to, for example, things like infection. And then lymphocytes, these are part of the adaptive immune system. They can acquire memory and they respond to other things like viruses. So all they did was they looked at the proportion of neutrophils to lymphocytes. And they plotted over this is what the population looks like. What I want to draw your attention is that actually within this population, actually there's a huge range of this ratio. Again, consistent with this idea that in healthy individuals with no known condition, there's a very large range in the ratio between these 2 cell types. And then when they looked at specifically the likelihood of dying from major adverse cardiovascular events. So this is basically, are these patients these healthy individuals likely to get things like strokes and heart attacks, and are they likely to die from them? And if you look at this here, is that actually your likelihood of dying from one of these events is 3 times higher than normal if you have a particular range of these this combination of neutrophils and lymphocytes. So there's now compelling evidence that even just measuring the total number of your white cells in your blood or the subcomponents of that is a very strong predictor of not just pathology, but also actually predictor of death. Now, zooming back out here, what I want to point out is that these are populations that were studied that were very heterogeneous, right? So what I mean by that, this is the people who were enrolled in these studies, range of ages, ethnicities, body weights, exercise regimes, genetic backgrounds. So what we've got a very diverse range of individuals, right? What this means is that we know there's a lot of biological variation amongst these individuals. There's a lot of noise, right, when we measure anything related to these individuals. What we are having, therefore, is a very heterogeneous population of people that we monitor over time. Despite that noise or heterogeneity, what we see is that actually, if I go back one slide, we can see that a simple parameter such as the total number of white cells is hugely predictive of the likelihood of them dying. What this tells me is that the effect of the immune system must be so huge that even when you take a very heterogeneous population of patients, the signal still comes through. Now, it is almost certain that for some people, the change in the immune system wasn't predictive of death. In some people, maybe it was actually reducing the likelihood of death. So for the whole population, this relationship holds. That means for some individuals, the effect of this relationship must be even greater. So when we average it all out, on average, individuals are 3 times more likely to die, for example, when we go from this end of the scale of the white cell count to this end. But for some individuals, the effect must be huge, must be 10-fold or 20-fold. And this, again, takes us back to this idea of actually stepping away from this binary category of normal versus abnormal, but understanding actually where is the individual phenotype for each one of us. And actually at the highest level, we don't yet need to almost worry about the intricacies of what is our CD4 T cells count, but actually what is the total number of our immune cells, and actually monitoring those. And almost certainly, it will be the case that actually as we start to deviate from what our baseline is, this signals an event that's taking place physiologically that is we should not be ignoring, and we should be looking at that. So how do we do that? Well, if you remember, I said as a doctor, I basically almost never requested a blood test on a person that has been normal, because we say, well, why do you order a blood test on somebody who is normal? There's nothing wrong with that. Well, unless we can start to measure people during health, during conditions of being, we'll never be able to really understand what their baseline is, so that we can't detect deviations. If we don't do that, then we'll fall back to what we have always done, which is really categorizing patients into normal and abnormal. Notice it sounds like I'm being critical of how we do things, and that is true. But what we've done historically with the resources that we had available was perhaps acceptable, right? But now actually we're moving into an era that we have the ability to generate the data and to also be able to monitor patients. So now we need to be thinking, okay, how do we now break away from this binary categorization of monitoring the immune system, and how do we now personalize that to individuals? So what are my take-home messages? So the immune system is absolutely critical for health and disease. I don't think this is a controversial statement, but critically, immune health is not a binary variable. In other words, it doesn't fit into normal versus abnormal. Those are extremes. When you are outside a population norm, then you're really quite sort of a huge deviation from probably your optimal system. But actually, the idea is that actually within the reference values, there can be significant effects of health. Personalized immune monitoring offers a paradigm shift in how we think about it, how we think about the immune system. Now, we won't know the effect of that until we start doing it. It's not unusual for experts even to think, well, there's no point measuring this particular analyte, whatever it is, because we don't know what it means. But the issue is that we will never know what it means unless we start doing longitudinal monitoring. And of course, that brings us to the work that Algocyte and Oxford Immunoalgorithmics have been doing. For us to be able to change this narrative, we need a number of technologies. We need machine learning, which we'll hear about from Jesper shortly, but also we need actually to be able to democratize the access to the blood samples and the blood tests so that actually we can do them at scale, we can do that for individuals over a period of time, and we can do it in a manner that's cost effective and actually it generates data that can be tracked over a period of time and then ultimately can be used to inform clinicians on how to make better decisions for their patients. Thank you for your attention.
So we now move from the immune system to the question of how we can look at the complex biological information and try to interpret them over time using the system medicine and AI. So our next speaker is Professor Jesper Tegnér, Director of Algocyte, Professor and Associate Dean of Biomedical Science Division at KAUST, with long-standing scientific links to Karolinska Institute, where he previously held a strategic chair professorship in computational medicine. His work spans system biology, computational medicine, and artificial intelligence. Today, he's going to talk about system medicine, causal AI for preventive healthcare. Jesper, floor is yours.
Thank you, Narcis, and thank you, Hector, and it's an honor to be here and talk to you today. So we have to take care of the immune system, we have to monitor it, and we have to move away from the binary classification as we just heard. So what I'm going to do today is I'm going to take you on a journey to show really the future is here in terms of what we can do with data, speaking of AI and machine learning in particular. I'm also going to show you one example of what you can do with blood. Blood is really exciting. And the third point is that the pioneering work that Hector started a number of years ago sits at the center of this convergence between AI and the blood. And the future is right here, as I said. So some of the topics here. So one word about me. So my only claim to fame is that I speak 2 languages. That is, on the one hand, quantitative languages, math, physics, and AI. And the other one being trained in medical school, so I speak the medical language. And that kind of characterizes the kind of work we do here. The second point, I've been fortunate enough to have a team of really skilled people. And when I say a team here, it also means I have a team of people with different mindsets. They are mathematicians, they are biologists, they're medical people. They are engineers, software engineers, machine learning people. So for example, I tend to hire people in computer vision, for example, and they do biology. And over the years, we've been doing this, and I have 3 people who have been enduring to work with me over the years, and 2 of them are here in the audience. Narcis, you met already. We've been working together for 15 years, and Hector as well. So thank you very much for that. So just one example of what we're doing. We are sort of trying to solve real problems, and we're a bit agnostic in kind of problems we're solving. It could be degenerative diseases, it could be immune-related diseases, or other kind of diseases, or we develop kind of tools for that. And we try to publish in sort of journals which sort of clinicians and biology people read. But we also do a fair amount of method development, AI, machine learning, computation kind of tools. And in essence, we are interested in the immune systems. I don't need to motivate that. We just heard a very good motivation for that, either as a driver of disease or responding to disease. The second part, we're very interested in the neurosystem, the brain in particular, from a medical perspective, but also as an inspiration for AI. So we develop tools for really understanding how cells are working in a fundamental sort of physical sense. And the second part is how cells are working in a context, in an organ, in a tissue. And that's particularly relevant if you think about immunology, you think about tumor immunology, for example. You have a number of different immune cells working together to fight sort of cancers. That's that. But you didn't come here to hear what I'm doing. So most of you know Hector as the super entrepreneur, starting a company, doing all this kind of extraordinary work. But this is not really how I know Hector. So what I did, I asked GPT, who is this guy really? And the response was actually quite good, I would say. So GPT knows Hector in the way I know him, that is as a nerd. Sort of working with complexity theory, developing new ways about thinking about these complex systems here. Gene regulatory network, fundamental complexity. And we also wrote the book together. Most of it, of course, was Hector writing the book. So together with Narcissus here on a new, sort of opening a new field of thinking about algorithmic complexity, not just a static kind of metric, but also a dynamic kind of metric to see how things are changing. So this is really Hector at the core. So he has this very fundamental kind of training and interest, and then having this entrepreneurial kind of twitch. And then I find the understatement of today, as you can read here, Hector is a bit sort of unusual in so many ways because he covers these different areas. And I think that is an asset, especially when you're doing this kind of work we're going to hear about today and the Algocyte work. So that's that. So we kind of transitioned from a lab doing 50% experiment to computation. Now we're entirely a computation kind of lab here. So the topics you can read about the program today, about AI, the blood cells, and the future and all that, I'm going to touch on that. So to give that some kind of structure, what we're going to talk about, I'm going to end about talking about healthcare and the blood and the position of that. But first off, I'm going to give a journey of AI here, segmented into bit historical part to give a perspective, and also how AI is currently developed over the last years in healthcare, medicine, and biology, and how fundamentally AI changes the way we think about how to do science and potentially change to do healthcare. Okay, so AI. Then when you think about AI and artificial intelligence, you have to think about the brain, right? And very much of the inspiration for AI comes from ideas in biology, brain included, of course. And today, most of us think about AI as a tool for looking at complicated data. The blood, it could be the immune cell, it could be the brain, or other kind of things here. That's true. But if you step back and look to the heroes of Hector, people started to think about this back in the 30s, 40s, and 50s, very, very early on. John von Neumann, I think, is a very good example. He was a very famous mathematician. He did pioneering work in quantum mechanics, but he thought particularly about two problems: the brain and thinking. What kind of language is in the brain? Is it mathematics or something different? And the second part, he was really interested in systems that could self-reproduce. And this was at a time when there was essentially no data, just clever people. And people started early on in the 40s to think about these neural networks. What is happening here? And the conception people had at the time was logic, of course, Aristotelian kind of logic. And also the third point here is the idea of how these systems are learning stuff. That is, how do they change? And Hebb came up with the idea, when you fire together, you wire together. And one point here is that most of these very fundamental ideas in this field comes from people sort of outside the field. Mathematician and Walter Pitts, he was trained in logic, and Donald Hebb, he was trained in psychology. And that is very typical for this field. So Hector, I think, is a good example of this, coming from sort of other areas into this area here. And so this was data poor, but then in the 50s, 60s, and 70s, people started to observe very interesting things in the brain here, because when you come from math, you may think that this is biology, everything is random and a bit noisy. No, it's not. So people studied the visual system in the cat very carefully and found specific neurons in the early visual cortex that were tuned to specific aspects of the visual scene. It could be an edge or a moving edge or some other kind of properties here. But these were all essentially observation patches, and people were thinking about how can this come about. But the bigger picture was kind of lacking. And the one who brought the bigger picture into thinking here was no other than Francis Crick, the co-discovery of the DNA helix. So he did his pioneering work on that. And then in 79, he wrote a paper thinking about the brain. So his point was that it's time to take these pieces and start to think, how do they come together and how does the brain really work? And at the time, if anyone else had said that, they would just say, okay, this is science fiction. But Crick, he's a smart guy. He was, of course, trained in physics initially, again, from the outside of the field. And his argument is that there could be no more important thing to study than the brain. And this is really the origin of taking pieces from biology to start to think about AI in a kind of serious way. And behold, people in computer science started to say that what happens if we take these kind of ideas from neuroscience and put them into a computer and try to train these systems? And that was the origin of what's called convolution neural networks, which has been used a lot to sort of find patterns in images. Now, there is a similarity between a computer scientist and a medical doctor. That is, both are extremely pragmatic. They like data, they like concrete things. So all of this could be just fantasies unless you have some data. And Fei-Fei Li realized this, and now we're talking about 2008, 2009, that to make progress, we need data, right? So what happens here, we collect a lot of different images, we give them labels, here's a cat, here's a dog, here's a table, and different variants of that. And then we train these systems, these sort of visual kind of systems on this data. And instead of having 100 engineers in the room, what is a chair? And all of us have different ideas, we try to write down the rules. We say, forget about that, and let's train on data instead. And behold, the whole field of computer vision threw out the old way of doing it and changed the whole game to neural networks about 10, 15 years ago. And the rest is history, as you said. So 2012, in a competition in computer science, they could beat all previous systems on image recognition and classification. But then you may say that, well, this is great, but who are interested in classification on cats and dogs? I mean, who cares? There must be something important we can do with this. And what was interesting here with these kind of network were that actually, when you look at inside these computer science kind of networks, the features, the properties inside these systems are very similar to when you do recordings in the brain. So this goes back to explainable AI. It's not enough you have to have a clever AI system that can classify something, but also you want to understand why is this sort of classified as a dog, what kind of properties, and these are the things you can actually inspect in these models to some degree. And that caught the excitement of the biology and medical kind of community. We can have explainable AI. So let me give you some recent examples of AI highlights here. And a good theme for that is always man versus machine. What can we do? And the initial conception was that everything we do, like vision and walking, is kind of simple, so people believe that is easy to do. But it's rather the opposite way. Things that we find difficult, math, chess, complex things, that's easy for a machine. And the things we find easy, recognizing an object, that's a hard thing for a machine to do. But games is a good starting point. And backgammon is a game that me and Flora likes to play. And in the 90s, this was one of the first example where using reinforcement learning, we can actually beat the best backgammon player. That's fine. I mean, that's a good prediction. But the more interesting fact was, how did a computer play the game? What can we learn from it? And it turned out that the computer was a bit more aggressive in playing the game as compared to the best human. So what happened next? Yes, the human sort of learn from the computer, maybe we should change the strategy to some extent. What was receiving much more attention was IBM's 1997 victory over Kasparov in chess. And to some degree, that was not that impressive because this was essentially brute force. So imagine all of you being an expert chess player. I go around and give me the best tricks. I plug them in the computer, and they have massive compute. This was what IBM essentially did. So it was not like the computer learned anything clever about it. So just brute force. So how do we move beyond brute force? And now we move to 2016. And this was, I think, a major event. So some of you may have heard of the game Go, which is much more complex than chess, many more combinations. So it's not just a game in Asia, it's more like a culture, a way of living. So you put kids in a sort of Go school when they're 3, 4 years old, and you learn to be humble and respectful and then play a game and thinking ahead and all that. And most of the people believe that the super master, Lee Sedol, he would beat this sort of deep mind system. That was expected. That did not happen. But what was more interesting was that in the second game, the computer made a move 37, and all the experts that commented the game said that, okay, AlphaGo made a mistake. This doesn't make sense. Aha, we humans are better. This is something wrong. But it turned out that this very odd move was exactly the reason why the computer could beat him in this particular game. So this indicates that there are ways of playing games that are novel to humans, even though we have been playing these games for decades, for hundreds of years, for thousands of years in Asia. So if you close the loop from Francis Crick to modern day, we see that thinking about the brain has given rise to new kind of AI. Most of the architectures comes from this biology kind of system, and we can solve specific tasks. All good. So what about healthcare and medicine and biology? So when we move domain from where we have games or YouTube videos, we don't have that much data. We have different kinds of data, and this is just one sort of illustration of healthcare data, and there's a lot of different sort of animals in here. You have EEG data, you have electronic health records, you have all different kinds of data in here. So the first mistake is to think that this is just one thing. No, it's not. It's much more complicated, and also having access to this kind of data. So it's a complicated landscape here. And if we look at the more coarse-grained level of this, we can see that we can develop tools for helping doctors to sort of doing administration, billing in healthcare, or we can also develop tools for patients to access their data and other kind of things. But the most interesting part, I would argue, is the sort of discovery part, using AI to discover new things about biology and medicine, and also drug development, which is a very sort of ongoing project in most of the pharma companies here. So looking back here, what kind of data are we talking about here? So before the Human Genome Project in the 90s, there was a lot of development of new tools for able to sequence on a large scale. And what happens over the next decades was a massive production of data. And this data production was not that now we're going to produce data to train an AI, but rather produce more data to understand biology better. Sequence data for the DNA and protein sequence data, and also different ways of tapping into cells, the activities into cells, and also image kind of data. So when you go back to, say, AlphaGo or classification of dogs and cats, you're going to ask, okay, we have all this data. What kind of tasks can be solved? And essentially two tasks were solved. One is sort of more famous due to the Nobel Prize, perhaps, solving the protein structure problem using this kind of sequence data and evolutionary kind of data, making use of all this data to solve a very specific, important problem. Less appreciated is the solution or a way of finding new antibiotics using these techniques. So you can actually search large chemical libraries and find new molecules in a much more unbiased way, these are, I think, good example of a real-world problem solved with these things here. So the key here is you have a task, you solve the problem, you have a computer science kind of thinking here, and then you use the data to that end. But zooming out, you can also think that you have this kind of system which learns a kind of language of life. So imagine you train this system, and the papers here are exactly papers doing that. So you feed this AI system with all these different kinds of data, and you train them, and it's almost like GPT. You have a sense of, it really understands what I'm asking about, because these systems sort of learn the language of life in this kind of way here. And this is exactly where we are right now. And also, these systems can also learn how to actually design an experiment, analyze data, design an experiment, look at the data, and close the loop. So we're seeing the emergence of virtual kind of labs, which sort of make contact with real labs here. So this is where we are right now. And just a few slides. A couple of weeks ago, I was in Cold Spring Harbor, which is one of the key center places for molecular biology since back in the 40s and 50s. And many people thought that this was the most exciting meeting ever at Cold Spring Harbor, so AI in biology. And it's kind of interesting to see that we move from sort of pattern recognition to more predictive, generative understanding, sort of causality, going from correlation to causality, and also very strong thread in these kind of presentations were the clinical application layer. So this is really where the field is going and trying to develop virtual patients, a copy in a computer of you, essentially. So we move away from this sort of simple finding some pattern in the data to making a model trained on data that understands you in the end here. Building virtual patients, building sort of tissue representation here, which I think is important for tumor immunology in particular, as one example here. So this is a notebook kind of summary, how we move from this sort of classical computational biology thinking to having models we can reason and interact with. So I think this is where we will see these kind of systems coming into healthcare, helping doctors, helping patients, and integrating different kinds of data. And we can develop these kind of systems that can eat the different kind of modalities here in a sort of data-driven way here. So I think this is exciting times for the future, and you can view AI as a sort of connective tissue across these layers. So today we mostly talking about the blood part, but the blood is sort of a very key module in the larger scheme, which can be combined with other data modality, and AI can clearly serve as a connective kind of tissue here, moving from individual molecules and specific enzymes to how cells are working, and also to image data here. So I think this is where the future will come here, and the blood will change that. So then we can think about this is really changing the way we do science, in the way that we go from, we have now these tools as an assistant when we do science, and we can discover new things with science. But the real test would be that if you train all these models with data up to some point, can we have a Newton, some new genius coming out of these computer models? This is William Blake's famous illustration of Newton discovering the secrets of the universe here. And we have people in the room here thinking about that, and no surprise, Hector, of course, been thinking about things. How can we think about this system to not only find patterns in the data, but also discover new things, to be creative, make fundamental kind of discoveries. So this is an interesting path forward here. So an interim summary of this AI part is really that when we have this kind of data and we train these systems of different kinds of data, be it games or be it healthcare data or molecular data, they will learn to solve the task in ways we did not expect. This is the essence of Move 37. We have solved the protein folding, but we don't really understand exactly how the system did it. We can sort of make processes in science very much automatic, but we don't have creativity or origin of questions still yet. And the same with healthcare. So can we use this tool to train and sort of solve personalized medicine in this way? So if we give them a well-specified task with a metric, can these systems figure out maybe new ways of looking at the data? And the immune cells, I think, is a good example of that, and finding new patterns in the data that can predict things. So I'm going to close with the blood part here. So how do we crack complex healthcare? And the naive expectation that many would have is that well, let's take all this data, make it accessible, and then we feed it into an AI and some magic will happen. I would say that this is a bad idea. So it's much more important to think not only of the data, but the kind of questions you're asking. And I think what you have in the blue here is generically true, but in particular for AI. So the less you know about the technology, the easier it is to be blindfolded and think that you can do almost anything with it. So you have to be careful, because depending on how we ask a question, we see what we sort of ask for. We tend to look in areas which we know, right? So we need to use these tools in a clever way and be precise about the kind of questions we're asking and the data we have, and using them as a companion here, right? So how do we transform this into healthcare? And this is something many of us can subscribe to, that in the eyes we have a window into the soul, and you actually have more in the eyes than that. We and others have done studies where you can actually say something about diabetes or different types of diseases depending on images of the eyes. So the eyes are kind of telling. And you can argue that blood clearly is, as we heard, a window into health, right? And this is not just science fiction, as we heard, because we have all these different immune cells doing different things here in complicated ways here. So the key idea here with this sampling is, let's sample the blood as a good entry point into personalized healthcare, and also as an important data type to tap into the healthcare system, not doing everything, but having more precise question and precise kind of data. That's the idea, to have some simplicity out of this complexity here. So last section here, so blood clearly is hot in several meanings, but yet, what can we tell from the blood? I mean, imagine it's like you put your ear to the ground and you try to listen to something far off being a disease somewhere in the body, and you look at the blood, which is quite some distance. What can you really tell? So many people have figured out that blood is clearly interesting. So competition is fierce, and we have a number of sort of studies coming out in these sort of good journals doing different things. One example is this one. And the point here is the kind of questions most of these papers are asking is kind of a different question. So for a physician, looking at all these different images is kind of tiresome, and trying to classify this image, which is good, which is bad, what is what here. So clearly AI, as this paper shows, can be very, very helpful in making this process much more automatic. But that doesn't mean we have improved healthcare. So many studies here, and this one, for example, are doing exactly this. But you want to say something about diseases. So what is the edge Algocyte have except having Hector in play here? I would say it's not just a software AI kind of layer on some data we access, but actually having a device that we can collect data in a systematic way over time. But what can we predict really here? So I want to give one example from a study we did a couple of years ago in the immunotherapy area. So this was one of the breakthroughs 10, 15 years ago. And it's kind of interesting. So I don't know if you heard about this guy, Jim Allison. So it kind of reminds about AI. It was a lot of hype with immune therapy. Maybe immune system could be good for something. And people hyped it, and then it didn't really work in the 60s and 70s, and people sort of run away from it. Then he started to look at this in the 90s, and he got the advice, as said here, don't do this. It's too complicated. I mean, there's so many cells, and how can we even try to tune these to diseases? So here is the loner in the room making progress. He got the Nobel Prize, and there's usually when you have the Nobel Prize, there's a sort of interview and some discussion, and I really recommend you to listen to that, because one of his patients was diagnosed with melanoma, and the doctor said, you will die within 3 to 4 months. No way. I mean, she had melanoma all over. And he tried his sort of CD4 kind of therapies on that patient, and she's still alive today after 5-10 years. And he was almost to tears. So this is really exciting. But it's complicated, right? Because we know that the immune system is complicated. We have variance between individuals, we have variance within an individual, and we have many players here, many different cells interacting here, and the cancers are usually clever because they want to cover, so they want to be invisible, so the Tregs, for example, cannot recognize them, etc. So how can we even do something here? And one of the major problems with most of these therapies is that some will respond and some will not. And these are not inexpensive therapies. So how can we tell who will respond and why will they respond and why don't these respond? So this is a very concrete question, but it's complicated for the reasons we heard in part before, that there are many different kinds of immune cells communicating together in different tissues against the particular kind of tumor we're talking about. So the very specific question, can we predict who will respond and not respond? And we could actually do that a couple of years ago in a paper here. So this was a teamwork with clinicians and all of that. And the important part here that we didn't need the whole internet of data, so to speak, only 8 patients for discovery and 20 for validation here. And this really says blood has a lot of information. So if you look at the data carefully and extract this information, you can actually make a prediction. So we could recover the markers. Many doctors were agree on that these are important markers for responders, non-responders, but we could also find new markers here. So what do we learn from this? We learn that even a few samples, we can do much. And this rich information in the blood here, and we can use these computational tools to extract these patterns, and we can predict something which seems very difficult, despite the complexity and the distance to the disease. So the advantage of the Algocyte, and also this study comes up, is the team. You need a team of different kind of investigators to do these kind of things. You need experts in medicine as well as sort of computation kind of tools. And what I particularly like with this image here is these two guys arguing here. And Narcissus and Hector know very well that we can argue about things for long times, and it's good to agree that we can disagree in a sort of constructive way. So the team is clearly a very important sort of key advantage here. But we still have a problem here. And the problem is we have a variability here. And there's a variability between patients, but also within a tumor. So how can we, by looking at the blood, say something? So we're kind of cheating with the responder, non-responder, because we had 2 extremes. We had 2 extreme labels here. They were far apart, so we could find some patterns. But what if we have a more continuum, and we have the variability in the baseline, as we just heard in the previous lecture here? And the key concept here, if you have a problem in space, you go to time, you look at things over time. So I asked, GPT, what is good with Algocyte here? And then, interestingly enough, GPT picked up that this is a very interesting aspect of the whole concept, that to look at things over time. Because even if there's a variability, as we heard, between individuals, you can see a pattern over time, even with sort of few data points here. And this is, of course, not a new idea. So if you ask a random clinician, give me one success story in medicine, many would point to this kind of very old study, Framingham Heart study. So it's old-fashioned in some way, but the key point here, you collect data over time, different kinds of data from the same individuals, and you follow them all the time. And then you can develop models that are very good in predicting risk and different kind of events here. So this is a key model to think about, and I was happy to see that you also cited the same kind of paper that I have here, a more modern example. So we can expect more data of this kind to come here. One of the pioneers is Leroy Hood, and it's a fascinating example, and of course he understood this very early, but this also gave a business kind of point here. So his idea was, okay, let's sample everything here from these individuals, have a deep sort of data collection here of different kinds and potential over time. That was good science, but it doesn't work as a business because it's almost like the name dropped me their first DNA company from Google, essentially. It's not a good, viable business model here because it's too expensive compared to the value here. So one has to be able to sample, have a good question, sample the relevant data, which is not too expensive, but still informative. You can extract information of in a good way to build a business. So that's that. So we've been talking about this, and my key takeaways are these. So for the AI part is really when we train these systems with huge amount of data, with a clear defined metric and a task, they will find ways of solving this task, which is unexpected. The move 37 again. I think that puts a very interesting perspective on the blood here. And we saw here that blood is a window, yes. So you can find a lot of information in blood. Blood plus AI, you can extract things. But if you add time to that, you can even extract more. And I would say, maybe Hector would not agree, we're just in the beginning here, because I don't think we have this kind of healthy control data in sufficient amount with disease data over time and looking at data in a more sophisticated way. I'm sure we will discover new kind of correlations we haven't really thought about. And if we have a clear metric and a task for this, defined as sort of personalized medicine here in a precise way, I'm sure if you train these systems on these data, we can find new ways of solving the task. Maybe we can have a move 37 in blood sitting here. So I end with that. Thank you.
Okay, we now move to the final talk, which bring together basically the idea of tracking biological signal over the time, algorithmic complexity interpretation, and disease signature for precision healthcare. Our final speaker is Dr. Zenil, founder and the CEO of Algocyte. He's associate professor in healthcare engineering at the School of Biomedical Engineering and Imaging Science at King College London, and also affiliated with King Institute for Artificial Intelligence. Please welcome Dr. Zenil.
Thank you very much. It is a pleasure to be here. I'm going to talk to you about the algorithmic future of medicine and blood testing with our new device that you see. It is at the center of the room. And we are very proud of our origins. I started thinking about this problem precisely at Karolinska Institute under Jesper mentoring and with Narcissa as my colleague almost 15 years ago. I think I still have a microscope in what was our common office. That basically what started Algocyte, started looking under the microscope, looking at individual cells. Because back then we were doing systems immunology. I was quite frustrated at seeing how far ahead we were with research, but how lagging behind we were with medical practice, especially primary healthcare, where you still go to the doctor and it is not data-driven at all. So then I moved back to Oxford as a senior researcher, doing research in AI applied to molecular biology. Moved to Cambridge, but before that, actually, I was approached by the incubator of the University of Oxford, and that's why we carry the name Oxford Immune Algorithmics. When I moved to Cambridge, I started looking at how to explore the space of possible hypotheses in the context of drug discovery, for example, for breast cancer. So we developed a lot of tools that eventually down the road is going to help continue developing the technology that we are bringing today to the market with this first product, which is what we call Algocyte Proxima. More recently, about 2.5 years ago, I moved to King's College, London, and I have my students actually working on some of these problems. And what motivates me is how we do healthcare today, how it is consumed, how it is provided, and how it is funded. And I call it a bricks and mortar approach to sick care, not even healthcare, because you only get medical attention when you are already ill, and basically you have to go somewhere else to get that attention. It is also very episodic and transactional. It is overcapacity, not only here in the UK, but this is a world problem. We have populations growing elderly, not training enough doctors and nurses, and this problem is getting worse over time. It is all population-based, just as Kourosh said. and this is a perfect way to close today, because basically the way I think of Algocyte is the AI meets the immune system, which is exactly the two fantastic talks that Kourosh and Jesper gave before me. And as I said, it's often not data-driven. Also, how it is funded, it is very reactive, and that leads to sometimes late diagnosis. And there's a lot of unequal standard of care. It's not the same living in London and having access to top care in large hospitals than living in the countryside or living in a developing country. And I think actually AI can make a difference in all these areas. Imagine in inequality, for example, if we had like a very good system that everybody had access to, basically everybody would get the same standard of care. So I think healthcare is the killer app of AI, AGI, or artificial general intelligence, or artificial superintelligence. And I think it is not me only or the Algocyte position, but you can see that every major AI company around the world is moving to healthcare. Take Oracle, for example, that bought Cerner. Cerner was the second largest EMR after Epic. So you would wonder what a database company is doing in healthcare. But take any other, Apple trying to release new wearables that are now clinical grade medical devices. Google, even Midjourney, that was kind of a surprise. The company that generates images is now getting into the MRI business with an ultrasound device. All others are also moving into this space because I think healthcare has the chance to actually be in some way solved with AI. That's the greatest challenge of humanity, how we deal with disease in the future. So the approach of Algocyte is basically to take the two pillars of modern diagnosis. And these two pillars, blood testing and medical imaging. Up to 70% of diagnosis goes in one way or another through blood testing. And medical imaging usually is used for confirmation, and there's some kind of overlapping. So actually, in our own technology, we use medical imaging for blood testing, because what we are doing is taking pictures of blood, uploading to the cloud, do all the magic with the AI that we have developed, and then come back with the results. So as Kourosh said, one of the main blood tests is a full blood count, as it is known in the UK, or a complete blood count, as it is known in the US. There are 4 billion of these tests performed, and over 95% of the time, when you are requested to do a blood test, it is included in that panel. So it is a huge market and a very important test. So this device that you see in the center of the room, that we call Algocyte Proxima, is able to perform a complete blood count. Today, we are releasing a device that is able to do a 3 differential. That basically means that we can recognize 3 different types of white blood cells. But very soon, and this is already in the pipeline, we are going to release one for 5-diff. So this is the first release of many others that are coming in the future. So in this brief talk, I'm going to be mixing a little bit of the vision of the company in the future and the actual device. I'm going to try to make an effort of what is available today and what is the vision in the future. So this test, as Kourosh said, is relevant to many diseases. Some of them very important are infection, anemia, cancer or cancer treatment, inflammation, and many others. This device goes beyond POC. POC means point of care. But beyond point of care actually is that you can place it actually at people's homes. So this is designed, and our end journey is actually to have this at people's homes. It is a rapid on-the-spot result, so you get results within 25 minutes. But we are also we will be releasing a version of it where you can get hemoglobin and hematocrit, for example, in 5 minutes only. And it is a best in class in its own way. So there are other 2 companies in the world trying to do this for at-home self-testing, but they only do a fraction of the analytes that we are capable of doing today. We do 13 analytes and more coming. The other ones only do a very small fraction. We have 2 independent sensors inside of the device. One is a spectrometer, and the other one is a bright field microscopy. And the advantage that gives us is that we can check one with the other. So with a bright field microscopy, we take pictures, and then our AI, which is both a convolutional neural network and a vision transformer, they come up with the results. But then if it doesn't match the expectation from the spectrometer, we can flag it. And today we do a version of that, but it's going to keep evolving over time to make it more robust. We have operated this device almost literally in the middle of the street. I mean, we went to India, obviously we were not in the middle of the street, but we had trouble connecting it to the wireless, and we were able to connect it through hotspot using a mobile phone on 4G, and it was still working. We have also tested it with a power bank. So that's why I say almost literally you can run a test in the middle of the road with this kind of device. And there's nothing like this on the market. Equivalent machines are much larger, more expensive, and more difficult to use. And this is completely user-centric. So this is controlled by a mobile phone. One way in which I explain all this is that actually we have brought professional medical instrument to the 21st century, because this is really how people today interact with things. With a mobile phone, through an app, it is connected through Bluetooth, and you can actually control the device with a phone. And you can see here some screens in which we walk the patient or the user through the whole test step by step. And this is what a clinician would get on the other end. They would have their patients, and they will be able to check the results. This is also something that is not very common. Usually what you have is a device placed somewhere else, and you have to print out the results. So this is one use case in which we think this is going to be incredibly valuable. And this is, for example, a cancer patient undergoing chemotherapy. These are 3 different pathways. At the top one, we have the patient that requires to get the chemotherapy scheduled exactly in the right time. And what happens today is that they go to the hospital, they get the blood test done, and if they are not ready because the immune system is too weak, they are sent back to home. So it is a waste of time for both sides, and basically they have to keep guessing when is the right time to give the next chemotherapy cycle. So obviously, if you are monitoring these patients more closely, you can make much better decisions, and the treatment can be much more effective. But another very important pathway is actually when cancer patients get an infection, and that can be very serious. And the problem is that the secondary effects of chemotherapy are very similar to the effects of an infection, or the symptoms of an infection. So they don't really know that they have an infection. They may not go to the hospital, get treated, and get antibiotics, and they may die in a couple of days out of sepsis, for example. So the third pathway is basically what you would have if the oncologist places one of these devices at patients' homes, and they monitor them every day, for example. They would create a baseline for that patient, and they will be able to make a better decision on whether someone has got infected or not. So potentially we can save a lot of high-cost events or reduce those high-cost events. So our idea is that oncologists are going to have like a fleet of these devices available, and they would distribute them with their patients for monitoring purposes. And the clinician has total administrative control over their fleet. And something that we are proving is that size and simplicity doesn't necessarily compromise performance. We have tested our own device against the Sysmex XN and then Siemens Advia. And whatever other POC devices stand for or promise in their own manuals, we are on average equivalent. And thanks to that, actually, we achieved regulatory compliance last week. Basically means that for some use cases, we can start rolling out this technology immediately. But at the same time, we are also evolving very fast. And I'm going to be telling you what is coming, because we want this at-home diagnostics lab to become into something that I call the health companion of the future. So we want to keep increasing the number of blood tests that we are able to do, eventually perhaps to cover most of the tests that diagnostics labs is able to run. We will be combining contextual and subjective data, and contextual means lifestyle. from wearables. And most of this, by the way, it is already done. It only requires to go through regulatory approvals. We are also combining it with subjective data, symptoms. So we create questionnaires that patients have to answer, and we enrich blood testing with all this information to come up with differential diagnosis. One key thing is, this is what Kourosh and Jesper have been talking about, is that we want to learn from the patient over time, build their own set point, and build personalized reference values that I'm going to touch upon in the next slides. One key thing is that we want to be able to build this kind of data. And you can see here that these are what we call disease signatures for different diseases and conditions, or even treatments. And they look very different. So I told you we are measuring 13 analytes in total. You are not seeing all of them because some of them, because this is a logarithmic scale, are at the bottom. But you can see how each disease may look completely different. So we have mapped all these to about 200 diseases, because that's exactly what we want to do. We want to flag for small deviations that start looking like into this disease or condition category. And that was the whole purpose of this device. We want to collect the data, then to do research. So this is a completely research and data-driven approach to blood testing. And as we speak, we are working on different projects and articles, some of them already out, some of them hopefully coming out soon. But this is a very interesting paper that came out a little over a year ago from a Harvard group in Nature that shows that basically each of us have different set point values. So we are not all similar to each other. Making the point of both Jesper and Kourosh, it is fundamental to understand each of us baselines to basically flag for these small deviations that currently we completely ignore into this binary categorization. So this is a paper of ours, and what we did was actually to look at reference intervals around the world and whether they made any sense. Because you would expect that perhaps people with the same genetics or at least ethnicity or in some other way would have like similar reference intervals. And it turns out that we failed to cluster by any measure. This suggests strongly that actually these have no biological meaning. That basically we have been taking these reference intervals almost for convenience from legacy systems. We controlled by a BMI, for example, and that made sense. We were able to cluster by ethnicity or region, for example. But when it came to reference values, it was impossible. So what we are proposing is to learn from the patient and the user over time their own personal reference values. So on top of using the population-based ones, we start narrowing down those reference intervals around your own optimal and flag for those small deviations. And again, this is not mock-ups. This is all done, and the technology that we want to push going through regulatory approvals. And you can see on the screen how we are taking population ranges, but also personal ranges. We also have proven an Algocyte score similar to what Kourosh showed for immune age. So this score goes from 0 to 10, the closest to 10, the healthier. And we also show that this was related to immune age. So if we got your chronological age right, it was that you were most likely healthy. And if it was off by a number of years, then you weren't. And we show these results with data from the CDC in the US, public data, and then with the UK Biobank here in the UK. And we are also, as I said, collecting a lot of data from multiple places. So we are reading wearables, for example, if you have a Dexcom for glucose monitoring, we are also able to put it all together, because what we want to do eventually is differential diagnosis. And we have this also uploading capability where if you get, for example, your blood test results from the NHS in PDF format, you just upload it. And by the way, I have the phone app myself and I do it all the time. So we are already collecting all this data from multiple sources. And as you can see, we can trace the source of any blood test result. You see the different colors at the top. So that means that, for example, the top one came from an OCR. So that was the user themselves taking a picture of a blood test result. So we make sure that all this is traceable. So imagine this deployed at a national level. Then you have all these scores around the country or different regions, and you can actually monitor people. And this is also key for clinical trials, for example. It's almost like checking the country's immune system for things like epidemics or pandemics. And this is related to what Jesper was talking about counterfactuals, because what we want to do is actually to build digital blood twins. So you can see on your left time series that are coming and generated from this device. Then we create correlation networks that become causal networks by looking into the literature, which is the work we did in Cambridge, for example. We create this kind of digital blood twin, and then you can actually play with this. You can do simulation of what would happen if we had done this differently, like given a treatment, a drug, or whatever it is. So coming back a little bit to reality, because I have shown you the vision of the company for the next, this is not months, probably years. We have already deployed this device in some places, including an NHS hospital for comparison study against another machine. And the lab technicians gave us this feedback, which is pretty good, on average 4.875. The only reason it was not 5 is because these technicians have run 500 tests over a year, and they would like to be able to insert the cartridge faster, for example, in the device, but it was not done for that purposes. It's not high throughput. And then we also took one of these devices to Canada, a community hub that is planning to use it for covering rural places. They have been using this device for a few months, and we got fantastic feedback from them, both on performance and usability. We only got slightly lower scores for things like integration and adoption, which is normal for new technology, and for training as well. And in that regard, for example, we are also trying to innovate in training. So we have created an app, which is an immersive experience, virtual reality, that you can actually test outside. So I invite you to go to the end where we started having coffee. We are going to have a session showing you the device and also the virtual reality, and we are going to have also drinks and nice snacks. But let me play this for those that have no time and may leave. Basically, you can place the device wherever you want. Remember, you are wearing this virtual reality headset, and you simulate the whole thing virtually. You even fingerprint yourself, and you saw the drop of blood, which was a red circle. And you can follow the whole test and run it virtually. This is a beta version, so it is still under development, but it is already quite nice if you look at it. So you deposit the drop of blood on the cartridge and then insert it into the device, and you get the results after a few minutes. So the way we explain the test is a combination of a glucose meter test, a sugar test, and a lateral flow test. So we require a very small amount of blood to run this test. And the idea is that we are going to have different cartridges for different purposes. I have these two with me, for example. So you can do hemoglobin and hematocrit on its own and get results within 5 minutes, or you do the whole blood count. And for every test, you would require a new sample. But the idea is that we keep covering more and more over time. And none of this would have been possible without the Algocyte team, that I'm very proud that a lot of them are here or are following online. Some of them have been with us almost 10 years. On average, I would say most of them around 5 years. So very strong core team that have been with us all this time. So let me run this video to show you what is the vision that we have for the future of this technology. And with that, we are going to close the day. I know it was a lot, and you deserve a break, and hopefully you are going to join us for drinks and snacks. And the three of us are going to be there to answer any questions. But let me run this.
Imagine having a medical companion at home, looking after your health and that of your loved ones. Introducing Algocyte Proxima and its Algocyte Health Hub. the first physical AI medical instrument that collects with minimal invasion a sample of your blood on a regular basis to build your health baseline. Then the Algocyte Health Hub will check your health against your lifestyle, collecting symptoms and monitoring key markers of your immune system. The future Algocyte Hub will learn from you over time and collecting all the data produced by the Algocyte ecosystem or beyond, allowing you to upload health reports from others with full traceability. The device can then compare your results with aggregated trends derived from millions of anonymized patient records and disease signatures to enable precise and predictive medicine.
From an at-home diagnostics lab to the health companion of the future.
Thank you very much.